Hi, my name is Sarah Whiteley. I'm a PhD student at the University of Canberra's Institute for Applied Ecology and the Australian National Wildlife Collection at the CSIRO, and I study the embryonic development of dragons. The purpose of my study was to understand the genetic underpinnings of sex reversal in the central bearded dragon, Pygona vidiceps. So we know that the species undergoes sex reversal, which is when a genetic male will turn into a female during embryonic development under the influence of high incubation temperatures. But what we didn't know was exactly how this process is happening at the genetic level during embryonic development. So in this study, I incubated eggs from the dragons at different temperatures and sampled the embryos at different developmental stages to sequence the developing gonads in order to understand the changes in gene expression that are associated with sex reversal. So what I found was that even though in the end both sex reversed females and normal females will end up as functioning females with differentiated ovaries, the ways in which that happens at the genetic level is very different between the two cues, whether that is a genetic cue or a temperature-driven cue. And we were particularly interested in the genes that were associated with sex reversal because it's such a novel process in reptiles and it's something that we didn't know anything about before. And I was able to show that there was a suite of genes that are actually conserved with other reptiles that have something called temperature-dependent sex determination, in which sex is determined by temperature with no chromosome influence at all. And these genes have really interesting implications for the evolution of thermosensitive sex determination systems in reptiles. This research is really critical because it provides a foundation of knowledge that we need in order to better understand the sex reversal system in the dragons. And we're planning on doing some functional experimentation where we try to manipulate the workings of the genes that we identified in this study in order to understand what, affecting the mechanism of that gene will do to sex reversal, with the goal ultimately to be to inhibit sex reversal under sex reversal inducing temperatures, and then also to induce sex reversal in the embryos in the absence of the sex reversing cue by manipulating the pathways that we've identified in this study.